Good morning, everyone. We're gonna get started. So, my name is Tatiana Trejos. I manage the Trace Evidence Facility at Florida International University in Miami. And our first speaker is Dr. John Goodpaster. Dr. John Goodpaster received a PhD in analytical chemistry and an MS in criminal justice from Michigan State University. Following postdoctoral studies at the National Institute of Standards and Technology, he served as a forensic chemist with the ATF laboratory. Dr. John Goodpaster is currently an assistant professor in the Forensic and Investigative Science program at Indiana University, Purdue University, Indianapolis. He teaches in the graduate program in the areas of alcohol and drug analysis, as well as trace evidence. And ongoing research in his group includes designing new techniques for explosives analysis, as well as instrumental and statistical association of trace evidence. So Dr. Goodpaster is going to be talking today about the analysis of trace evidence using microspectrophotometry and multivariate statistics.
Okay, thank you very much, and congratulations everybody, you woke up yet again for our final day. It's been a great meeting. I've really enjoyed being here, and I've learned a lot, and I've actually changed my talk a little bit to reflect some of the discussions that we've had over the last couple days. So without further ado, I'll just launch right into this. This is what I'm going to talk about first, is just some general philosophical statements about the nature of class evidence, which is what we're dealing with when we're looking at trace evidence, typically. And then also the role of chemometrics, the use of multivariate statistics to look at items that have class characteristics. And then I'm going to highlight 3 different applications. All of them have in common both a chemometric approach and the use of microspectrophotometry, which is a very popular instrumental technique in trace labs all across the world. So here's the reality. It's a wonderful reality, isn't it? You've got a sample, somebody else got it for you, they send it through the mail. You don't know where it's from, it's usually small, it's usually all beat up, and you have no idea how it was deposited. But I guess the good news is, on the other hand, you have an exemplar that's usually bigger, that you do know where it's from, and it's your job to compare those two things. So how can we realistically and reliably approach this type of problem? So this is from my ATF days, and I'm just showing you some Qs and some Ks. And by some of your standards, my Qs are beautiful. They're huge. I mean, we're not talking about like a sliver of a paint in a tape lift or something. So, but you can see that Qs are scorched. They're dirty. They're embedded in a putty like the second one. And so how do you do a phthalate comparison between smokeless powders if one of them is already covered in phthalate because it's in a putty material? Or down at the bottom, a plastic Easter egg. How would you approach a plastic Easter egg comparison? Is there a database of yellow plastic Easter eggs out there that I could consult? Not really. So there's a lot of interesting problems when you're facing these Q and K comparisons. All right, so I'm going to propose some steps. The first step, and I discussed this with our class on Monday with Steve Morgan in that workshop, is understanding class evidence and what it is and what it's not. So in order for it to be useful, it has to be 1, widespread, 2, diverse, and 3, reliably differentiable. So we've got to see it in our cases and therefore care about it. We have to know that there's different types of whatever this item is, and we have to have a means to differentiate the different types. So that's should be pretty clear. But we also accept and expect and readily admit that at some level, items of class evidence are going to be indistinguishable. We're not going to be able to narrow it down to one plastic Easter egg out of all the plastic Easter eggs in the world, something like that. So we just know that going in, that there's going to be, at some point, these become indistinguishable from one another. Now, this is what I call forensic logic. And this gets at the aspect of when we do a QK comparison, exclusions are pretty solid. But it's the tough part that really gray area is when we have an inclusion. So, and that's based on a kind of a logical argument that if you're given a Q and a K, as I show at the top of this slide, the first thing you do have to assume is that they're representative. So at some point, you know, your Q and your K can represent a common source. And then you go about trying to prove that fact. So you've got 2 logical statements. Statement A is that Q and K originate from the same source. All right, so statement B is that they have indistinguishable characteristics. The only thing that we can address is statement B. All right, because if we say if A then B, we have to assume that if they came from the same place, we're going to have the same characteristics. We've got to have that happen in order for our approach to work. But the only thing we can address is B. We evaluate B. If B is not true, then we can say, by logic, if not B, then not A. We've excluded that sample. All right, it does not have the same characteristics, therefore they cannot have come from the same place. But unfortunately, this is just the way it is that you can't say if B, then A. Just because they have the same characteristics, we cannot say, and we never would say, that they therefore did come from the same source to exclude all others. And that actually just follows from logic. If you have a statement, if A, then B, you can reverse it. You can say, if not B, then not A, but you can't do the final step. You can't say, if B, then A. And then step 3, this is what Jay Siegel, my mentor and the director at IUPUI, says all the time. So don't oversell the evidence, but give it its due. So with the exclusion, we can definitely exclude. With the association, there's all sorts of ways to say an association, and I won't bog down in that. It's all about what words we like, what words we're comfortable with, but you have to say something about the fact that it could have come from another source. Just because they're the same doesn't mean they came from the same place. So it's up to us to figure out significance. How do we articulate the significance of an association between two samples? And ideally, to do that, we would know something about the sample, how many classes there are and how reliably we can differentiate them. So how do we do that? This is just 3 examples. There's a number of different ways to approach this problem. I've done a lot of the first one, where you do class discrimination studies, take a number of samples, see how much differentiation you see within a certain population. That's number 1. Another one that we've seen here this week is also pairwise studies. So collect a number of samples, analyze them by multiple techniques, and at the end of that analysis, decide how many of these pairs of samples are left indistinguishable. And that reflects the extent to which we can differentiate. And then lastly, environmental studies, as I call them, are classic examples of target fiber study, where you take a particular sample and you say, how much of this sample do I see when I just sample the environment? So how much of a random chance is there that I'm going to find a particular sample? So I'm going to dwell on the first one, because that's where a lot of the chemometric techniques that I've done are very helpful. So the comprehensive approach. And I harped on this in our class on Monday on chemometrics, and this is what I like to do, is that put this approach up and point out that only number 4 has anything to do with mathematics. Okay, so number 1 is delving into the sample, understanding its manufacture, its distribution, that kind of thing. Then going out and gathering your samples with that amount of insight in terms of how it's distributed. Then looking at multiple techniques, not necessarily the one that you like the best, but evaluating different ones. And then finally applying some sort of quantitative measures to differentiation. And then unfortunately for trace, these populations are changing over time, as we've discussed. So you do need to keep up with those changes by monitoring them over time. In terms of chemometrics and what it can do, you can break that up into 2 main types, supervised and unsupervised. And I'll talk about both types today. So unsupervised does not impose any structure on the data, it just reveals the structure. So you do not tell any of these algorithms how the classes are organized, it just attempts to see that organization. Supervised is the reverse of that. In that case, you actually tell the algorithm, these are the groups of samples that I believe exist, tell me how reliable this is. All right, to what extent are these classes actually differentiable? Here's a typical procedure. So you go through some mathematical pretreatments, and I'm going to talk about 3 main techniques today: clustering, PCA, and discriminant analysis. Now I've got 1 slide on each of these, and it's hopeless to try to explain multivariate stats in 5 minutes, but I'm going to just give you a qualitative view of what these techniques do. So in terms of the clustering analysis, what that does do is make up classes. So it studies the similarity of your objects, and it will define for you an organization. And it'll form that in terms of a dendrogram. And I've shown just a generic dendrogram here in terms of how it's divided up into branches of different types of samples. And you can tell that, for example, samples A and B are rather different than samples D, E, and C, okay, because they end up on both different branches of this large dendrogram. With PCA, we're looking at correlations. Instead of just basic similarity dissimilarity, we're actually looking at correlations between variables. And we're looking to try to find some underlying factors that help to explain these correlations. All right, and so the main benefit of PCA is that you can actually visualize complex data like a spectrum or a chromatogram in 2 or 3 dimensions. And then you can use your brain just to see those patterns, more so than you would be able to by sitting in front of hundreds of pages of spectra and trying to figure it out. So in this case, if you've got a set of 6 samples in 2 dimensions, this is a very simple example. But if you have that, you can say, all right, looks like there's a correlation here between the X variable and the Y variable. As X goes up, Y goes up for these samples. So if you do a PCA on this kind of data, what you would see is that the principal component would be drawn along an axis that explains this variation. So you'd have a principal component right there. All right, so it would explain the variation along this axis, because that's the axis that is most variable for this particular data set. Now, principal component 2, we would continue to do this. It would find the next largest source of variation, or the next largest axis, and in this case, it's pretty simple. It's just the right angle to principal component 1, and it just describes the width, or the scatter from principal component 1. Then last but not least is discriminant analysis. We've got known groups. This is a supervised technique, so you tell the algorithm how the data is organized, and you ask the algorithm to spit back some reliability. How reliably can I differentiate these different groups? So you train it. It can learn in a way, and so you train it with some known samples, and then you give it some new data, and you ask it, where does this new data fall in? So if we just have a simple example of 6 samples, sample G, which just flew in from underneath, is put in there, and the algorithm says, you know what, sample G looks a whole lot like samples D, E, and F, and with high probability, it belongs in this group. And that's essentially what we're doing when we do trace evidence comparisons. So we are assigning observations to groups and deciding whether or not they both belong into the same group. All right, so what's in it for us? I've made a list of what chemometrics can and cannot do. And this is relevant to the stuff that we've been talking about this week. It can do a lot of great stuff. On the left, it can help you see groupings. It can help you see organization in a group of samples, and it can tell you something about the reliability of those groups. And so that's a very useful thing to be able to do. It also can help you find characteristics, like areas of a spectrum, for example, that are really distinguishing, that help differentiate different samples. And ultimately, there are some ways where we can associate knowns and unknowns to the same group. So using discriminant analysis, and that's something that I've done in the past with different samples. What it cannot do, you're not going to be able to type in something and it's going to spit back 1.4% error rate that you can testify to. That's not going to happen. Because most of our forensic protocols involve some qualitative steps and quantitative steps. Chemometrics can take multivariate quantitative data and do something with it. And I'm not aware of a chemometric technique that can somehow automate or discriminate based on a visual or stereo or comparison or PLM, something like that. And ultimately, the ultimate question of deciding whether or not they came from the same source, you're not going to be able to punch in some data and a little green light comes on and it says they're from the same source. And then a little red light says, no, they're not. I know of no technique that's going to do that. So the problem, and I'm just being really honest with you here, is that chemometrics is hard. I don't know if you guys remember that Barbie from Mattel got in trouble because they pulled the string on the back of the Barbie doll, and then she said math is hard, and it caused a ruckus because it reinforced the stereotype, 1, that math is hard, and that 2, that women would have trouble with math. So it was actually very controversial, but you could pull the back of a John Goodpaster doll, and he's going to say, chemometrics is hard, because it is. It's non-intuitive in many ways. And so how can we go about being comfortable with these techniques if we don't understand them? And so, also, are we having statisticians trying to usurp our role? Are we taking the human judgment, putting it on a shelf somewhere, and using computers to make decisions? All right, so that's my rant stage, I guess, my soapbox stage about chemometrics. And so what I'd like to talk about next is microspectrophotometry and chemometrics. And in particular, what can chemometrics do for a microspec kind of experiment? So in terms of UVVis microspectrophotometry, I'm not going to dwell on this because I know a lot of people have these instruments, but it's nice because it combines both a microscope and a spectrometer. So you get a microscopic view of your sample as well as spectral information about that sample. And so I'll be talking about 3 different types of materials, all of which have some sort of absorbance in the UV or the visible. And here's just a picture of our instrument. I'm sure plenty of you guys have maybe even the same instrument, but they essentially are combining the two so you both have an imaging portion of your instrument as well as a monochromator so you can resolve absorbance wavelengths. Okay, so part one, analysis of dyed hair. This was an idea that came along before I came to IUPUI, but essentially that microspec is used for dyed fibers all the time. But what about dyed hair? You can definitely note the fact that hair is dyed under the microscope, but what if you treated it like a fiber? Can you actually do a spectral analysis on a piece of hair that's been dyed with a particular type of hair dye? In terms of what this looks like, the downside is that hair protein absorbs beautifully in the UV, and so you're always going to have a contribution of the hair protein. But in the case of red hair dyes, you do see a nice hump or an absorption here that's due to the dye. And there's two images there. One's showing a dyed hair, the other's showing the same hair that's just been extracted. So just the difference between the dyed hair and a bleached hair, okay, which is a pretty extreme example. And so we approached this using those techniques. So the first is, this is the Dendogram showing the clustering analysis of a number of different dyes. So this is all the same person's hair. The only difference is it's been dyed with all these different dyes. And you see on the left axis, that's the list of 50-some different hair dyes. All of them are nominally red, but they're all also nominally different by different manufacturers. And you can see, too, that there's three main groups. There's this dotted line that is cutting this Dendogram to the point where you see three main branches. And at each branch, I'm showing you the picture of what's called the central object, which is sort of the poster child of that branch. And we can use our brains, and I would say to you that you can see that there's class number 1 and class number 2 are pretty close in terms of their color, whereas class number 3 is clearly different. And if you look at this Dendogram, that's what's going on. You see the branch that incorporates class number 3 splits off at quite a big distance from class numbers 1 and 2. And so you can immediately tell that we've got two types of hair dye or two groups of hair dye that are relatively close. The third is quite a bit more faint and less intense. And on the right are the spectra. So it's also undyed hair, so you can just see what hair protein looks like, but then it has class 1s, 2, and 3, the spectra of each one of those. And you can see that the level of absorption along the spectrum of the hair correlates with the perceived color. So class 3, which is rather faint, has relatively low absorbance, and so on. Now, next step is PCA. What we should see is a PCA plot in 2 dimensions where each one of these data points is a spectrum, and it should agree. We should see 3 main groups. So what I'm showing you here is a PCA plot where the classes have been color-coded according to the designations that I just showed you. So there's class 1, 2, and 3. And in this case, they appear to dwell in 3 distinct areas of this plot. And so that's a good thing. You want your unsupervised techniques to agree with one another, and that means PCA and cluster analysis. Now, the added benefit of PCA is it'll help you look at these spectra with a new eye. It'll be able to say what parts of the spectrum are more important than others in terms of describing variability. Okay, so in this case, we're looking at what's called a factor loading as a function of wavelength, and where you see high values of factor loadings, those are areas or wavelengths that are highly correlated with these principal components, and therefore merit further inspection. This is where you want to go back to your original data and say, okay, what's going on in this part of the spectrum? Why is it that these regions seem to have more correlations? Okay, so here's the major areas of principal component 1, and so we're in the 250s, 300s, 450s, 550s, somewhere in there. And then here's principal component 2. Now, if you superimpose this on the original data, here's principal component 1, and you'll notice it's honing in on the leading edge of the absorption of the hair protein. And it's also honing in on where the dye absorbs. Okay, so those 2 regions of these spectra are highly correlated with this principal component, and they explain a lot of the differences or a lot of the variation between samples. If you overlay principal component 2, you'll see that it covers that trough, if you will, between the 2 major absorbances as well as the dye area. Okay, so this would make sense, that if this is something that you were to stare at these spectra, you'd say, okay, it looks like I've got 2 main features. Those features would largely explain what's going on here. But the added benefit of doing PCA is that it can do it in a quantitative fashion and visualize it for you. Okay, so it does something for you that would take you more time or might even be impossible. Then last but not least is something called discriminant analysis. And in this case, it actually says, all right, if you really have 3 classes, how well can I resolve them? And here are the 3 classes, and it reports an accuracy. It actually gives you a number, and it says that for these 3 classes, 89% of the time, these observations were correctly classified. And so then you can say, you can feel either good or not so good about that. The higher the number, the better you feel. But this is an example where you can actually say, all right, so there's 11% of the time there's confusion going on. There's errors being made. What classes and what samples, what dyes are getting confused with what other dyes? And you can look this all up by going through a discriminant analysis approach. So concluding this, we definitely classified hair dyes. This is being written up, and part 1 is somewhere in the editorial process of the Journal of Forensic Science. But we definitely saw that, and we saw regions that were more important than others. We did say, by the way, that you can't classify by brand. There's no way we're going to take a piece of hair and say, yeah, it's Clairol. Yeah, it's just not going to happen. And we tested that by using discriminant analysis. We can actually say, if you try to do that, your accuracy goes way down. And so it's simply not feasible for this data. All right, part 2 is Clearcoats, and we've done a lot of work with this, including with some international collaborators. So we're looking at, again, at the Clearcoat, the top coat of paint, and it has a lot of UV absorbance, and so we're using microspectrophotometry for that. Here's the Dendogram. Again, looking at 3 classes, that's just a coincidence in this case. We just have another three groupings, one that's largely apart from the others. But you also will see that class 1 seems to split into 2 separate subclasses beyond that. Here's the objects, central objects, and you'll notice there appears to be some correlations going on here. If you look at the peak on the left, as it goes up, the peak on the right goes down. So that would imply correlation. And that's what PCA is going to hone in on. It's going to say, there's something going on between these 2 regions. So we would expect to see, when I look at these factor loadings, that those regions of the spectrum are important. So here's our 3 groups in PCA, split out, and they all seem to dwell to some extent in their own place in this 2-dimensional plot. There's obviously some crossing over, so these classes do mix at the boundaries. And here are the factor loadings. If you recall from before the correlations between the different areas, I'm going to just advance ahead and show you that. That's indeed where PCA is finding these differences. All right, so it's going to be correlating with those 2 main features that seem to be going up and down in concert. And then last but not least is the discriminant analysis. In this case, it's 93%. So if we assign 3 main groups to automotive clear coats in our sample database, our accuracy is 93%. The ability to use discriminant analysis to get more detail is shown here. So you don't just have to say I'm such and such percent accurate. You can actually delve into this more and say, all right, what 2 classes are most confused? How many observations of this class are assigned to that one by mistake? Well, not by mistake, but just by an inappropriate classification scheme. And so this is a matrix showing along the diagonal the correct classifications. Those are all in green. All the numbers in red represent observations that were wrong. So they were originally assigned as class 1, but then discriminant analysis went back through and assign them as class 2. So you need to take a look at those samples, find out why is it that those are being confused between classes. And so summarizing part 2, we do have some organization. We've identified the features. And we also were able, just like with hair dyes, if you try to assign trends or classes based on make, model, and year, it doesn't work. And it can provide you that answer. It's good to have negative information. And so the classification by make, model, and year were not reliably observed. Then last, and also this is the newest project, is looking at dyed cotton fibers. And it's beautiful for microspec because there's not much going on in terms of bulk chemical composition, but the color of the fiber makes a big difference. And so we've started this project off, and Steve Morgan and I are going to start collaborating on this kind of project. And we've just done the same kind of approach. And again, if you take a look at these central objects, the pictures are worth 1,000 dendrograms, really. I mean, when you look at the 2 pictures there, class 1 and class 2, those look a lot alike in terms of their hue, whereas class 3 is quite a bit different. And that's not an error, that's the way it is. And it's nice to be able to visualize that in a structured dendrogram. If you look at the central objects, you also notice that class 3, the spectrum of class 3, is showing a shift relative to class 1 and 2. And just backing up, you'll notice that class 3 is the one that looks a more vibrant red. So spectrally, it's also different. And so we can use these spectra as exemplars. We can look at the correlations of the wavelengths. And this is all making sense in terms of how the data is organized. Then last but not least is discriminant analysis. So Goodpaster can differentiate dyed fibers with 100% accuracy. What's wrong with that picture? Well, I think the obvious answer is we've only looked at 3 different dyes. All right, so that's the ultimate challenge with these types of studies. So I could claim that I could differentiate these 3 dyes with 100% accuracy, but what about all the other red dyes that we haven't looked at yet? So that's going to introduce the opportunity for more overlap between classes. And then wrapping this up, we can discriminate those 3. Is this representative of all red cotton fibers? No, clearly not. So we need to do more work in terms of looking at the different red dyes. All right, so I just want to acknowledge all the people that played a role in this. So Elisa Lazuski was here on Monday helping us out, and she's done a lot of the work on the dyed cotton fibers and Clearcoats. The Indiana State Police spurred a lot of these projects. So we've been working with their microanalysis unit on a lot of these different studies. We got some free hair from a local beauty college for all that hair dye work. And then also Julie Barrett, who did the hair dye work, received a graduate thesis grant. So I thank you very much, and I can answer any questions if we have time.
Is there any questions? Is there any questions?
I have one. Morning, John. Bill Davis, Harris County Medical Examiner's Office. I was just wondering, was there any pre-processing of this data, or was it just raw absorbance versus wavelengths?
All of it was pre-processed. So the biggest thing that we did was take out baseline. So we did a baseline correction and normalized. Because as you know, when you do microspec along a fiber, you'll get high absorbance and low absorbance, and that kind of variation will just naturally occur. So if you normalize, all of that goes away. So we're just looking at normalized spectra.
Thank you.
I also have a question for you. How many hair samples do you have to take in order to take into account heterogeneity on the hair? How many hair specimens you have to take to account for heterogeneity now?
Okay, yeah. What we've been doing with these projects is try to conform to guidelines of the trace evidence community. So for example, on the cotton fibers, we look at the fiber guidelines for how many spectra, and right now we're doing 10 spectra per fiber. With the hairs, we went with 20 hairs as a simulated K to look at intra-head variability. And then we also did some studies where we took 20 hairs that were all dyed with the same hair dye and then randomly picked 1 hair to be our simulated Q and tried to see how well can we link those 2 together. Yeah.